Hey everyone, Jack here, and today I'm going to take you through B cells. B cells are such an important cell in the adaptive immune system, so I'm actually going to start with them and then I'm going to jump into the slightly more complicated T cells in the next videos. So let's just jump into it. What does a B cell look like in the blood? This is just a regular H&E blood stain, and we can't really tell a B cell. What we can tell is a lymphocyte. So these neutrophils, monocytes, basophils, eosinophils are quite easy to spot. A lymphocyte is just a large nuclear cell with not a lot of cytoplasm here, but it is still a white blood cell compared to the red blood cell here. Now they're called lymphocytes because you typically find them in lymph nodes, but we also find them in the blood, and we can see that it's a lymphocyte, which means it's either a B cell or a T cell. We can't tell whether it's a B cell or a T cell though, when it's a naive lymphocyte in the blood. So here's our B cell. I'm going to use them in green for these presentations. And now B cells, their defining feature is that they're coated in an antibody. Now that antibody is unique to that B cell. So each B cell in your body has its own antibody, and it will express just one kind of antibody all over its cells. And that antibody has very high affinity for pretty much a single antigen. And so antigens will bind to the antibody. Now they're called antigens because antigens generate antibodies. So they're antibody generators. So you put that word together, you get antigens, right? And so antigens will bind to the antibody that is on the surface of the B cell. And in this case, it's acting as a receptor. So we call it a B cell receptor. And it really is just an antibody that's embedded in the membrane of the B cell. So here we go, we've got an antigen here. Now, hopefully, ideally, in a healthy situation, the antigen will be a pathogen antigen. So it will come from a bacteria, a virus, a fungus, or a nematode, or any sort of pathogen. They'll be releasing proteins, but also it can be other biological molecules, and these will bind to the antigen. Now, if it was a protein, typically antigens, somewhere in the binding region of the antigen is somewhere between 7 and 25 amino acids. So it's actually really short when you consider most proteins are in the hundreds or thousands of amino acids. So we're talking about just binding to part of a protein from a pathogen. And it will bind to just this antigen. So this antibody on this B cell will bind to just this antigen. And each B cell was covered in about 10,000 of these receptors or antibodies. Now, if we zoom right in here, we can see that it's bound to what's called the variable region of the antibody. And that's because the whole antibody isn't completely different between each B cell. It's just a variable region, which is the antigen binding region, which differs between each B cell and each antibody. So the variable antigen binding region is unique to each B cell. But here, there's an interesting problem here, and I love this problem. And I'm an innate immunologist, but I must admit this is where the adaptive immune system gets pretty freaking cool, right? So here's the problem. The human genome contains around about 27,000 protein coding genes. Those genes contain the recipe for 27,000 proteins. But B cell receptors, antibodies, are proteins, right? And there are roughly 10 to the power of 11 or 10 to the power of 12. There are a hecton for sure of different antibodies in your body, right? So these antibodies have different amino acid sequence, and that amino acid sequence should be described by a different nucleotide sequence, so a different gene, right? So how is it that we have so many different antibodies that are proteins that should be coded by a different sequence of DNA and thus a different gene, but we only have 27,000 genes? So what's going on? We obviously don't have a gene for each antibody. Something weird is going on. And the solution is when, the adaptive immune system starts to look really cool. The solution is self-genetic engineering. Basically, the adaptive immune cells self-genetically engineer to induce variability in their own genome. So let's see how that might work. Here we have the gene for an antibody, and it's present in a pro B cell. This is not a mature B cell, it's a pro B cell. Now, the gene for the antibody is actually made up of lots of little chunks, and each of these chunks are sort of a functional unit. Lots of little chunks, right? And what's going to happen is the pro B cell is going to self-genetically engineer and only incorporate a few dozen potential chunks. It's actually close to 70 or 80 potential chunks could go into the final antibody. And it's only going to incorporate a few of these variable chunks into the antibody. And then there'll still be this part of the antibody gene will be present in all antibodies. And that's because this part corresponds to the non-variable region of the antibody, which is the same in between all B cells. And this part of the antibody gene here corresponds to the variable region of the antibody. Now you might say, how do you get that much variation from only a few chunks? Well, it's a lot like a poker hand, right? So if I deal out a poker hand, I've got 52 different cards. And if I deal out a poker hand of 5 cards, there's actually 2.5 million different ways that those cards could be dealt out as 5 cards, right? And it's exactly the same. You could imagine these little units of this DNA as like a card in the pack, and we just deal a new hand for each B cell. Each B cell gets its own little shuffled 5-card hand from which it creates its antibody. So the pro B cell then goes to a pre B cell, and the pre B cell then gets checked to see, does this antibody bind to a self antigen, right? So like I've got loads of proteins in my body. I don't want to produce an antibody for that because that antibody will disrupt the function of those proteins. And this is what causes autoimmunity, is when we produce antibodies for our own proteins. So we definitely don't want that, right? So a pre-B cell then gets checked to make sure that the antibodies don't bind to a self-antigen. And once it passes that check, it then goes on to become a mature B cell, a mature naive B cell, because it's not active. Now we call this process somatic recombination. Now somatic just means it's not part of our germline, it's not producing sperm or eggs. And recombination is very common when we produce sperm or eggs. It's a way we generate genetic variation for the next generation. But recombination is very rare in somatic cells that aren't to do with sexual reproduction. And so it's called somatic recombination because it's a very special kind of recombination. It's very unusual for recombination to occur in somatic cells. But that really just means, honestly, it just means self-genetic engineering, because this isn't the mRNA, this is the DNA. It has lost its genetic sequence is now different to another B cell, right? It has a unique genome only in these 3 genes that are involved in producing antibodies, but it does. This B cell has a unique genome compared to the rest of your body, which is amazing, right? We're told that every cell contains the same recipe book, but it's not quite true in the adaptive immune system, right? These cells contain a unique couple of pages in their recipe book that is their genome. Now, how does this occur? Well, in between each little chunk, which is like a playing card, each little chunk of this DNA that makes up an antibody, there is a recombination signal sequence, right? And this is a little small 12 or 23 nucleotide sequence that exists in between these chunks. And they are recognized by an enzyme, recombinase, you could call it, which essentially binds to the recombination signal sequences at random, brings them together, chops them and glues them, right? And when you do that process, there's actually a lot of proteins and enzymes involved in this process, but when you do that process, that is how you genetically engineer yourself. Now, the key enzymes here, we call them RAG genes, so recombination activating genes, because they're the genes that are active in recombination. And we have RAG1, RAG2 in the human body, and they bind together to form this complex that does the chopping to create the recombination. So RAG1 and RAG2, they recognize these signal sequences that exist in between the units of the antibody that we're going to chop and change, right? Cool. So now we've got this unique antibody that's formed from self-genetic engineering. What happens after an antigen binds to it, right? And a lot happens, and it's very, very cool. So let's go through it step by step. We have Well, the first thing that happens is if the antigen is attached to a pathogen, right? So here we have what's probably, in this case, let's call it the SARS-CoV-2 virus, COVID-19, and it has those spike proteins, right? And let's say this antibody binds to those spike proteins in SARS-CoV-2. What will then happen is the B cell will phagocytose. And that's quite interesting because we normally think about the innate immune system when we talk about phagocytosis, but the B cell does it too. So after that B cell receptor, which is just an antibody, is activated, the whole pathogen will get phagocytosed. A lysosome will fuse with that phagosome to form the phagolysosome. This will then go to the Golgi where there's an MHC2 molecule. Now you might see where this is going. It's going towards antigen presentation. So the components of the virus are now broken down by those lysosomal digestive enzymes, and a fragment of a protein might bind to the MHC2 molecule in the Golgi. Now this will then go into a vesicle, which will then go fuse with the cell wall, and now we have an MHC2 antigen presenting molecule with a little bit of a pathogen, a little protein from the pathogen being presented outwards out of the cell. Now what's very important here is this antibody will bind to one antigen site, but it may not be the thing that's being exposed on the MHC2 molecule. And that's very interesting, and I'll come back to that a little bit later. Right, boop, boop, boom. So now we've got a piece of the pathogen here bound to the antibody, and you can see it's colored here to represent the antigen that will bind to the antibody. But you can see over here, we've got the blue piece. We've got a different fragment from this protein that's come off a pathogen being exposed on an MHC2 molecule. So what happens next? This is where the T cell comes into it. So it's called a T helper cell, and I'm going to do a big dive into T cells later in a later video. But a T helper cell will come along, and guess what? It's got the T cell receptor on its surface, and that T cell receptor has undergone a similar genetic shuffling that the antibody went through, right? So the T cell has also self-genetically engineered itself so that the T cell receptor, TCR, is completely unique to this T cell, right? So the T cell receptor here is completely unique to this T cell, and it stumbles along. So multiple T cells might come along and go, no, I don't recognize the antigen that you're presenting on this MHC2 molecule. I don't recognize it, and nothing will happen. But eventually it will find that one T cell that goes, hang on a minute, I recognize this. And when this T cell receptor is activated in response to the antigen that's being presented on the MHC2 molecule that came from the pathogen that the B cell ate, and it ate that pathogen because it recognized its antigen, a different antigen. Once it recognizes it, the T cell is going to release a cytokine called the interleukin 4, IL-4, and that cytokine is going to go bind to the IL-4 receptor on that B cell, and it's going to activate it. Now, what you can see here is we've got this kind of checkpoint, right? What if this was a self-antigen, right? Maybe this accidentally was a self-antigen. Let's say it was part of the cartilage of my joints, right? And if we end up with an immune response to the cartilage of my joints, we might end up with rheumatoid arthritis, which is an autoimmunity to the cartilage inside your joints, right? But let's say you're a healthy person who doesn't have that autoimmune disease. What might happen is if this is the collagen in your joints, it will go inside and then it will produce a different fragment onto your onto the MHC2 molecule. It's very unlikely, right? First of all, it's very unlikely that the B cell would survive that selection process inside the bone marrow, because normally things that recognize a self-antigen are killed. Normally B cells that recognize a self-antigen are killed, right? So it's incredibly unlikely that we end up there. But let's say we did. Let's say that checkpoint failed, and now we've got a B cell that does recognize our joint collagen, you know, the cartilage in our joints. It's also very unlikely that a T cell, they go through the same selection process. What's the probability that a T cell that's going through this selection process would also make it through that checkpoint, even though it recognizes a self-antigen? And what is the probability that this T cell would also survive that selection process? If this was 1 in a million, and this was 1 in a million, surviving that safety check, now you've got, 1 in a billion chance of having that response to that antigen. We do have a lot of self-antigens, so 1 in a billion might start to shrink those statistics as we have a lot of self-antigens. But you can see that by having this 2-stage activation requirement, it helps put the brakes on autoimmunity. Right, so the T cell has now activated the B cell with IL-4. What happens next, right? I guess we always say what happens next. So we've got this 3 signals that have now happened. An antigen has bound to the antibody. A different antigen has been presented by an MHC2 molecule and been recognized by a T cell. And then we have IL-4 has been produced by a T cell to activate it. So we've got 1, 2, 3 things going on that have all happened to this B cell. And after we get all 3 things happening, we then get division of the B cell. Now, this B cell will just pretty much just divide. It will go out into the lymph nodes and divide. This is one of the reasons why your lymph nodes swell up when you get sick, and you'll get division in the lymph nodes. And yeah, so that's the first thing. It's essentially just division of this exact same cell. Now, why would you do that? Well, remember, this cell has a unique DNA sequence because it's self-genetically engineered, right? So if we now copy it and copy it and copy it and copy it, we have more clones of that unique genetic sequence that recognizes that antigen, right? So it has now expanded up. So now we have more of these B cells around ready to respond if we got exposed to this antigen and this pathogen that expresses this antigen again. So a good example is this antigen could be the spike protein on SARS-CoV-2. But that's just creating memory cells. That's dealing for a future problem. How do we deal with the problem at hand right now, which is a pathogen that has been recognized by both B cells and T cells? And so this is the real guts of what's going on here. We get the effector cells. So we get B memory cells and we get B effector cells. Now in the clinic, we would call those plasma cells, but they're really just B effector cells. Now the big difference here is they will have a bigger cytosol, right? Their cytosol will swell, and that's because the cytoplasm is full of rough ER. So we get an expansion of rough endoplasmic reticulum. Why? Because we're now going to produce a bucket load of protein, and that protein is going to be soluble antibodies. So not antibodies stuck in the membrane of the B cell, but secreted antibodies. So they are going to produce a bucket load of antibodies. And this is where the rubber meets the road of the B cell. Now, the memory cells are predominantly going to live in the lymph nodes throughout your body, whereas the plasma cells are going to go everywhere. They're going to go in the circulation, they're going to go into the tissue, and that's because that's where you need those antibodies. You need those everywhere. But what do antibodies do? And what I might do is actually pause here, and I will cover what antibodies do in the next video.